This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 703, recorded on January 6th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. It's a pretty nice day here, although it's a little chilly. 37 Fahrenheit, 3 Celsius.
Also joining us from Austin, Texas, Rich Condit.
Hello, everybody. We got 64 degrees and occasional rain, sometimes heavy. It'll clear mid-afternoon with a predicted high of 64 and sunny. So, you know, typical Austin winter day.
And from Southeastern.
Your winter doesn't require a TWiV hoodie.
Yeah,
that's correct.
Brienne's chilly. Don't you control your heat, Brienne?
I do, but for some reason I was just chilly today.
I can see the thermostat on the wall behind you, actually, right? Is that
yes,
correct?
That is correct.
All right. And from Southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's 34 Fahrenheit, which is 1.111111 Celsius. And we have what I was told by a TWiV listener that a Michigan friend told them is our Michigan winter unicloud. We just have a cloud for the whole week, the whole winter month, whatever. But yeah, cloudy.
Well, here we are in a new year, and it's zipping by already. The first week is by, and before you know it, it'll be the end of the year. Hopefully it'll be a little better, but things are not going so well at the moment, as everyone knows, in many, many fronts. Nevertheless, we, what is it? We soldier on? Is that the phrase?
Yeah.
We soldier on. I just want to remind you that the Aaron Diamond AIDS Research Center at Columbia University is still looking for a BSL-3 assistant manager. We'll put a link for that in the show notes. And what's up with ASV, Kathy?
Well, as you've already been hearing, our abstract deadline for the meeting that's in July is February 1st. And so you need to start working on your abstract. It's only 26 days from when this drops tomorrow.
Kathy, many people are saying that in-person conferences will be greatly curtailed for the future. I think ASV and ASM will have in-person meetings again, right?
I think that we will. The platform that we're using is actually able to do hybrid meetings, and so that may be something of the future, but we certainly didn't want to try for that this coming year.
Yeah.
We didn't really think people would be traveling, and it was going to be more complicated than just having an online meeting.
So you think in the future maybe you have both an in-person meeting and that will be then also broadcast for people who want to watch?
I don't know. Maybe. You know, there is this added benefit that people from around the world can come. The disadvantage is that we're still going to be doing it on some time zone in North America, and
right.
that may not be convenient for around the world. But we're going to have recordings available to people who register for about a week. So if we continue to do something like that, maybe we will do hybrid. But I just imagine trying to do, you know, these kinds of things in every room of, for instance, the Monona Terrace.
Yes.
You know, it's
Yeah.
mind-boggling.
Yeah, it's not easy. The time zone thing is a real consideration. But, you know, I teach my classes online, and some of the students are in China and Singapore, and they say we have to deal with it. We don't have any choice. So, I'm going to start teaching my virology course next week, and I think Kathy and Brienne are also teaching. And this is the course that I started a year ago, and it was interrupted, as everyone else's was. So this time it's all online. Very different, which I'm sad about because I like going downtown and, you know, seeing students and so forth. But we'll get back to that someday. I don't
Yeah,
know
we
when.
have a couple of in-class hands-on activities that we're not going to be able to do, so that's disappointing.
Some of the Columbia classes are actually hybrid. There's part in class and part online. You know, you have to keep the class density very low. And so I give my lecture in a big lecture hall, and it would only fit by distancing standards like 40 students. And normally it sits hundreds, right? And so my class is 130 students this year, so I can't do that. So we'll do it all online.
We have some classes that are going to be online and some that are going to be hybrid and a few that are going to be in person. So I'm teaching a lab section that's going to be in person, but I have been making a list and thinking of all sorts of changes I need to make because of some of our COVID restrictions. That's kind of been taking up my time lately. So I'm going to have students staying in their lab stations, and if they want to put a tube in the centrifuge, they have to raise their hand and go with no other students around them within 6 feet and things like that. So it's taking some interesting planning.
Yes, for
So
sure.
your sabbatical is over.
My sabbatical is over.
Was that just a one semester deal?
It was, yes. So it was one semester where I could do some research and not have any teaching. Although, of course, 2020 research was a bit different than normal.
Right.
I never took a sabbatical. Did Rich or Kathy? Did you guys ever take one?
Yeah, I went to Phil Sharp's lab.
That's right.
In 1986 for about 10 months, and that was a game changer for everybody.
Did you take one, Kathy?
I've taken 2 in situ sabbaticals, kind of for personal reasons. I needed to or wanted to stay here, but I didn't have to do teaching or go to faculty meetings or etc. So I had more time for other things, and I did kind of go to a couple places or take a couple of extra. Like I took a statistics thing, and so I did some things.
Yeah, I never did. Never went anywhere. Never took an in situ sabbatical. Well, some people would argue that I'm been on sabbatical for the last 15 years. But I wanted to, in fact, related to that, I forgot to thank someone last week on our New Year's Day TWiV. I have to thank Amy Rosenfeld. We did thank her for the space station episode, but you know, she came to my lab 5 years ago and I said, I need the podcast. Can you run the lab? And she's been doing that very well, writing grants, papers, doing all the experiments. So I have to thank her for that because it lets me do this. Otherwise, I wouldn't have a lab because I would not be able to keep it funded on my own. And I do enjoy having it. It's not going to be forever, but thank you for taking care of the lab while I get distracted by this and other things as well. And the reason I mention it is because it's not in my job description to do this. In fact, it would be hard to find any academic place, especially a medical center, where you could spend a lot of time doing science communication. It just hasn't been worked in. One exception, though, is University of Utah, where Janet Iwasa is a science communicator, and that's what she does. She doesn't do lab work, and they arranged a tenure thing based on what she did, which is brilliant, right? We're going to have her on in the next few months, and she can talk about that. And it can be done, but, you know, at my place, you need to write grants, and that's it. And at a medical school, that's typical. So I'm able to do this because I have someone who can run the lab, basically.
This science communication thing has acquired a special significance twist for me in the last few days, or I've been thinking about it differently. Have you seen the, what is it, New York Magazine article?
Yeah.
And, well, that guy's career, the author is a fiction writer.
Yeah, Nicholson Baker, right? Yeah.
And, you know, he's actually done quite a lot of research for that article.
Yeah.
Okay, and he's identified all of the players and a lot of the events and that kind of stuff. But what occurs to me in the long run is that he has, and I don't want to sound, you know, like science is some big deal that, you know, is an exclusive thing, but he doesn't have the scientific training to be able to evaluate what he's reading on his own. Okay? And I think in order to do really effective science communication, you have to be able to understand the science. And that takes a special skill because, it's not a trivial matter to actually understand the science in sufficient depth to be able to evaluate all that stuff and communicate it at the same time. So it's a tricky field.
So I was contacted by a fact checker for that article a couple of weeks ago, and you may have been contacted by fact checkers before. They want to check to make sure that statements made in articles are in fact right, and not everybody does this. You need some time to do it. So she called me up, and she had sent me the article, and I said, in my opinion, this is science fiction. If you want me to comment on that, I'm happy to. And she was silent. Fact checkers typically are very straightforward people checking facts, right? I said, never mind. I'll be happy to go over the facts with you. I interspersed it with my opinion, right? But none of that comes out in the article. All they want to know is was this bat virus isolated in 2013 in a cave out, that kind of stuff. So, I tried my hardest to say this is nonsense, but they published it anyway because they know people will look at it, unfortunately. But I think it's science fiction, yeah. All right, we have two papers to chat with you, but first I wanted to chat about a conversation that's been ongoing for some time, and we've gotten a bunch of letters about it. And that is this idea of, hey, why don't we just give everyone one vaccine, one shot of the Moderna or Pfizer vaccines, and that way we could immunize more people. And this first came on my radar because Michael Minna actually, I think he wrote an op-ed in the Times a couple of weeks ago about this idea. And we mentioned it at some previous TWiV and said, no, this is a bad idea, because at the time we had some Pfizer data about efficacy of the vaccine after one inoculation. It was a very small number of people, and it was 52%. So we said, this is a crazy idea. Then I got letters saying, it's not right for you to say it's crazy. You should look at the data. And then in the meanwhile, Pfizer came out with more numbers. So now, as Kathy points out, this is moot because the FDA yesterday said it's a bad idea to just do one shot. We have data that says we need two, and there's a good statement by the FDA which has come out saying it's not a good idea.
That's a really good statement.
Go ahead,
Brianne.
Yeah, I was just going to say, I do think it's a little bit less moot just because there is still quite a bit of discussion about this, and this is occurring in other countries. This is occurring in the UK. And, there still are some very strong proponents of it here. I would be a very strong opponent. I have pretty strong feelings about this one. And I don't think it's a good idea. Or at least I don't think it's a good idea based on the data that we currently have. So I don't think it's quite as much a total moot point, but I do really like the FDA statement, and I'm pleased that they pushed back.
Well, what I look forward to here is an informed discussion so that the listeners can understand what the issues really are, and myself included.
All right. So the new stuff has been added since this morning. So there's the FDA statement, and then also SLUI and Warp Speed have said things. So let's put that at the end after we just chat about the science a bit, okay? And we'll come back to that. So Michael Minna on December 18th in an op-ed said we might be able to do one dose. And his statement was based on the Moderna data, which came out, I think came out about that time, but I forgot the timing. But I was only aware of the Pfizer data. And I went back to the two documents that these two companies submitted to the FDA, and there's one table in each one about this. And in the Pfizer one, their estimated vaccine efficacy against confirmed COVID-1 after dose 1 was 82%. I'm sorry, was 52.4%. All right. But it was only a few individuals. So, in fact, I think I have the table there, right? Isn't that it? Yeah.
No, you don't have the table. Where?
The table
Pfizer?
is for Moderna.
Moderna table is
Pfizer.
further down.
Oh,
So there are two tables I put in here. They're both from the Moderna report.
Right.
Yeah, okay. And the Pfizer actually, I just put the percentage, the vaccine efficacy from Pfizer. So estimated vaccine efficacy, 52.4% against confirmed COVID occurring after dose 1 but before dose 2. All right. And the confidence interval was 29 to 68%. Big range there because there are not a lot of people involved in that.
So what's the first part of this statement? I was confused by this, Vincent, because the first part of the statement on the Google Doc reads, says after dose 1 was 82%.
Yeah. I think that's
then,
anything after dose 1, which might include dose 2, right? So if you average dose 1 and dose 2, right?
Okay.
But just dose 1 only is 52%.
Dose 1 only is 52%. Okay.
And that's why I said originally Minna's idea was not a good one. But then I don't know if the Pfizer data the Moderna data had come out or not at that time. But now we have them. It's in the report for the FDA, and I probably overlooked it, so my error. But we have Table 15, vaccine efficacy of Moderna's vaccine to prevent COVID-19 from dose 1 in patients who only receive 1 dose. Okay, so only 1 dose. And they're split into 2 groups. You have dose 1, 1 to 14 days after dose 1, and then more than 14 days after dose 1. And in the one, so you get your first dose, and then 1 to 14 days after that, some people got confirmed COVID, obviously, and the numbers are pretty small. In the vaccine group, 5 out of 1,000, and in the placebo group, 11 out of 1,000, which is a vaccine efficacy of 50%. So 14 days is clearly not enough time. However, if you look more than 14 days after dose 1, then 2 out of 983 people in the vaccine group had confirmed COVID, and 28 out of 1,059 in the placebo group, which is a vaccine efficacy of 92%. And that, I think, is where Minna and many other people who have commented on that said, hey, it looks good after dose 1, right?
Still very small numbers.
Yeah. I mean, it's 2,000 here, which is more than the Pfizer, but yes. It's
but it's only 30 cases, okay? And as I recall, that they didn't want to make a call on this initially until they had at least twice that many and more like 3 or 4 times that many.
Yeah, considerably more than that. I think it was in the hundreds that they wanted.
So I would call that, promising, but...
Yeah.
Exactly. That, I can certainly see the argument, but I'm not sure that we have the data to support it at this point.
Yeah, I think you would need more data. I mean, part of the issue is, as you all know, a lot of vaccine is being distributed and not even put into any arm, right? They're sitting in places because of logistics. So it's not clear to me that this would solve that problem at all, right?
Yeah.
We know nothing about the longevity after 1 versus 2, right?
Right, exactly. So to me, there are sort of 2 pieces to this. One of them is that they've looked, after dose 1 for a relatively short period of time in a smaller number of people. And so we don't know anything about longevity, and we don't know whether this would require people to get vaccinated perhaps more frequently than if they had actually gotten the originally designed dose. The other thing that sort of concerns me that's on that sort of idea is that if you look at the original data that have been published for both Pfizer and Moderna in some of their phase 1 and phase 2 studies, they showed antibody titers after both the prime and the boost. And there was a very dramatic difference in antibody titer.
after
that boost. The boost did quite a lot. There is a document, a link that I put in here that I guess we'll talk about later, that the UK used for making this decision about having one dose of the AstraZeneca vaccine. And again, I look at the data, and they have neutralizing antibody data after one dose and after two doses, and they are dramatically different. And so I sort of, sure, we don't know the correlates of protection. And so maybe that lower level of neutralizing antibodies is sufficient. But based on the difference that I've seen from that boost, and based on sort of the low numbers and smaller amount of time with the data that we have here from Moderna, I'm not sure that we need to change recommendations quite yet.
So one of the things that is in this article that I put in this morning is that as Dr. Mascola of the NIH Vaccine Research Center says, we've always known that the amount of vaccine has been an issue. And if you think back, Tony Fauci and others were saying, it might not be till next fall or toward the end of this year, 2021, that we're in a position where we've got a majority of people vaccinated. And this is all the more reason why multiple vaccine platforms are going to be needed, because just the manufacturing capability is tremendous. And this article also reminds us that the Johnson & Johnson vaccine only requires one dose, but we haven't seen the efficacy data, and they haven't applied for an EUA or anything yet. And also, regarding evaluation of the existing data from the phase 3 trial, it was always planned, evidently, for Moderna and the NIH Vaccine Research Center to go back and look at that data and evaluate it, and also look at some of the phase 2 data. And so this article spells out sort of the things that would need to be done to provide the FDA with additional data to perhaps go forward with just one dose. So look back at the patients from phase 2, or conduct a new trial to demonstrate that those that got the 50 microgram dose developed a threshold immune response. So this is talking about a 50 microgram dose versus a 100 microgram dose. So this is a different way of stretching the amount of vaccine that's needed, for example.
Yeah. So I have a question, and I don't know the answer, but as you know, when a new therapy is approved, and then you want to test something else that you think might be better, you cannot withhold that approved therapy, right? It's called standard of care. So is it an EUA considered standard of care? Because one, it is not.
I don't think so. Because
then if you're under EUA, then you could say, okay, we're going to have a placebo group that gets nothing, even though we know that we have 2 other vaccines that seem to be work. So an EUA does not fall under standard of care. Because that was my question. Right?
I don't know, but I don't see anything that has indicated that would be the case. I mean, I think these articles would point out that would be a complication before the other approval of the biologics license application, the BLA.
So I would like to hear a wrap from Brianne on what a boost is.
Okay.
I want to know what it does, and I think I know enough now so that I can understand what you say. Not that I couldn't before. Understand better. I want to know what it does, and I'm interested also in scheduling. If you look at the childhood vaccination schedule, for in the U.S., the timing of the boost is variable depending on the vaccine, put it that way. So what does a boost do? What do you know about timing?
Sure. So what does a boost do is actually a little bit easier of a question. So when you have a B or T cell, first see its antigen, it will make a type of response that's known as a primary response. So it will go from being naive to start to undergo a program where it gets better at its functions, it proliferates, and changes into basically something called an effector cell. And eventually, most of those effector cells go away. They undergo apoptosis. It's a good thing. You don't want to turn into a walking lymph node with all of the T cells you had from your first infection ever. Then you wouldn't have any room for the T cells responding to your second infection. And you are left with a group of cells that are memory cells. Those cells are better able to respond when they see the antigen. So they respond both quantitatively better, they divide faster, and they also act qualitatively better. So they can do a lot more. And each time you see the antigen, you sort of get an improvement in those memory cells. So you can get a secondary response the second time and a tertiary response the third time. And each time those memory cells will be getting better at what they are doing, and will be sort of increasing to get to, maybe some level that might be the threshold for protection. So the boost is giving you that exposure for your memory cells in order to allow them to have a second round of seeing the antigen to help those cells differentiate into good memory cells, a lot of them, memory cells that are potentially making better antibodies in the case of B cells, things like that.
So is the improvement just in affinity maturation? So you have a better antibody, or is there an effect also on, say, the size of the pool of the memory cells or how readily they respond?
All of the above.
Okay.
So with the B cells, you will see changes in the affinity, so the affinity maturation. You will partially also see some class switch, at least as the response goes on. Doesn't necessarily have to be a memory cell. There are more cells than there were before the response, and those cells have changed their ability to respond.
So how do I decide when I'm going to do a boost, and can I wait too long?
So I have looked into a lot of this, and if you look in Plotkin's vaccines textbook, which lays out some of this quite well, it is very clearly stated that 3 weeks after the primary inoculation, the primary response has reduced enough so that you have a boost. So the idea with a boost is you need to wait until the primary response is done. So you don't just have the effector cells get rid of your boost. You actually need to be working with the memory cells. So
at least 3 weeks.
At least 3 weeks is that definition, yes.
So how about the other end?
So I have never seen a question of too long. And I will say that, the experiments I did as a graduate student, I gave my boost to 8 weeks, always. But in the situation that we are currently in, we want to get you into that boosted state as early as possible. And so you can see that's why Pfizer is going at that first earliest time point, 3 weeks, in order to get you. Yeah.
The reason I ask is that now this is just hypothetical. I'm just talking. I'm not advocating anything. But if one were to, say, alter the distribution protocol, one potential consequence of that is that people get their first shot and then get boosted instead of at 3 weeks or 4 weeks, say 2 or 3 months down the road, or even longer, 6 months. And my question is, in terms of the effectiveness of the boost, I'm not talking about whether or not the primary vaccination would be effective, but in terms of the effectiveness of the boost, is there a downside to that? I don't know the answer to that. I
think that there probably is a sweet spot. And I think that sweet spot is probably going to vary from vaccine to vaccine. As you mentioned very well, the currently approved vaccines have different amounts of times for that boost. I could certainly imagine something a little later being good, but I just would want to see the data on that. Because I think that there probably, like I said, is a sweet spot. There could be a place where it is a bit too long, and there could be a place where it seems to work quite well.
And in terms of these vaccines, we probably don't know. We don't know what that sweet spot is. They've waited at least 3 or 4 weeks, so they know they're going to get a boost. And probably the strategy is to make it quick enough so that you get everybody boosted to maximum potential as soon as possible.
Right. That is my thinking as well. They may have some of that information done in animal studies that they haven't published, but we haven't seen any data on that.
Great. Thank you.
Someone who's a proponent of stretching out and having the boost be later could potentially argue that if in the meantime someone got the actual infection, that would serve as a proxy boost, as it were, and if their protection was from their initial inoculation with the vaccine was enough to give them some response, that might reduce the severity of their disease. But that's all quite hypothetical, I think.
Yeah,
sure. I
I
think that makes sense.
These tables that we referred to about one dose, they didn't actually look. They didn't have severe COVID. It was just COVID. So we don't really know about severe prevention of severe disease after one dose. Just
right.
So I think all of these discussions are interesting, okay, and they're not wacko. It seems to me, but I'm with the FDA. FDA is following the science, and as I said on a previous episode, every time I think about FDA and approvals, I think about thalidomide, which, I'll find an appropriate link to refresh people's memories on that story sometime.
Right. So this thing that was mentioned about Moderna and the Vaccine Research Center looking into their data is actually not the idea of delaying the boost, but instead reducing the initial inoculation and the amount in it, and therefore stretching the amount of available vaccine and still getting the benefit of the boost that Brianne has pointed out is so important.
Yeah, and that actually, there's slightly more data, or at least slightly more reasons to think about that, both from what Moderna and the VRC are saying, and if you actually compare the Pfizer and Moderna vaccines, they are different doses, and Moderna's dose is quite a bit higher than the Pfizer dose.
Right. And they say that it could take, I can't remember if I already said this, the research could take about 2 months, and then it would have to get approved by the FDA.
So Saad Omer, a vaccine expert at Yale, said in a Times article, "We have an issue with distribution, not the number of doses. Doubling the doses doesn't double your capacity to give doses." So I always come back to that, where, as Daniel said, 80% of the doses are sitting in freezers, and we're not giving. So this doesn't solve that. We still have to figure out how to get those to people. I notice now that CNN has a vaccine distribution counter up on its news, right? They have all the cases always and deaths. Now they've added vaccines shipped and vaccines given in the upper right-hand part of the thing. So I think that's an important thing. If you tomorrow said, yeah, we can just do one dose, it's not going to solve the issue. There are bigger issues of logistics that have to deal with.
Yep. To me, that's one of the most important pieces of all of this, is that I'm not sure that these proposals completely solve some of the problems that we're having. I also think about the conversations that I have with people who are having questions about vaccines. And I know that when I answer them, I talk about the fact that I have felt confident in the process and have felt confident in the trials process and the science. And so to me, if we're going to make good on those arguments, we should follow what was done in the trials process and follow what was worked out. And so if we have some additional data, I am happy to think about some of these alternative scenarios. But right now, when I'm trying to talk about vaccines with people who don't have a ton of background on this, I want to be able to use data to support what I'm saying. And right now the data we have is with the trials done with 2 doses.
I have confidence in the FDA on this, and my thought has always been from the beginning of this discussion about 1 or 2 doses. The EUA is for 2 doses, and you may be seeing something different in the data, but the FDA decided that the 2 doses was the way to go. So we'll link to their statement, which was released on January 4th. I want to read a few sentences. It's a very well done, clear statement. We have been following the discussions and news reports about reducing the number of doses. And I must say that, a lot of this has been amplified in the news, quoting different scientists who have different opinions. Extending the length of time between doses, changing the dose, half dose, or mixing and matching vaccines to immunize more people. These are all reasonable questions to consider and evaluate in clinical trials. However, at this time, suggesting changes to the FDA-authorized dosing or schedules is premature and not rooted solidly in the available evidence. Without appropriate data supporting such changes in vaccine administration, we run a significant risk of placing public health at risk, undermining the historic vaccination efforts to protect the population from COVID-19. And then they go on to say, what we have seen is that the data in the firm's submission regarding the first dose is commonly being misinterpreted. In phase 3 trials, 98 participants in the Pfizer trial and 92% of participants in the Moderna trial received 2 doses at either 3 or 4 week interval. Those participants who did not receive 2 doses at either a 3 or 4 week interval were generally only followed for a short period of time, such that we cannot conclude anything definitive about the depth or duration of protection after a single dose. From the single dose percentages reported by the company. So even though there were 2,000 people, right, with just 1 dose, they're saying we didn't follow them long enough. Anyway, so it's a good statement. I really like it. I think it's well done.
The article I was referring to was in the New York Times. It's on the front page by 2 women journalists, Cheryl Stolberg and Sharon Lafreniere, who I had not noticed before, but I think they did a good job of spelling all this out too.
So what's going on with Slowey, Kathy? You put some quotes from him.
That's from this article.
He's advocating the half dose thing. I was shocked at that. I couldn't believe he's doing it because that's he essentially at least in some capacity represents the administration, right? I mean he's running warp speed, and so to me that's a message that's not consistent with the FDA. That it's confusing. Okay,
so both he and Fauci are confirming this research, looking further into the phase 2 data and potentially extending that. But Slowey went further, saying that basically that the FDA and Moderna are already discussing the possibility.
So he says that the 50 microgram doses showed an identical immune response to 100, but that's not what we're looking at. We're looking at efficacy, which is different, right?
Yeah.
Mm-hmm.
That's the important parameter. Are people protected? And that just as I think Brianne said earlier, it doesn't matter if the immune response is, so you got to know which one is protecting you. So
all of these ideas, or some of them, might be right.
Yeah.
Okay, but we don't have the data.
And basically what you said, Vincent, is exactly what the next paragraph says Fauci says.
Oh, sorry. I didn't. Yeah. Yeah, I'm glad when I'm online with Tony Fauci. It means,
it's good.
Good thing. It's good. All right, good. So for those of you who didn't like our treatment of Michael Minda's idea, that's the science behind it. And, in this pandemic, things get amplified because people are afraid. And I think the FDA is taking the right position. We have to go on the science and how we approved it. They had that committee, those two committee meetings to look at the data for a whole day. And this is what they decided. And I'm okay with that.
I'm really glad that the FDA, responded to what's going on in the press and put together a nice statement like this.
So what's going on in the UK? They're using Pfizer vaccine as well, right? So what are they doing? Does anyone know?
They're using Pfizer vaccine, and they've also approved the AstraZeneca Chadox.
Yeah.
And they are going with 1 dose in order to stretch their number of doses. There are even reports of people having their scheduled second doses canceled so that those doses can be given to others.
So I thought the AstraZeneca was originally approved in the UK based on 2 full doses, right?
I believe that's the case.
Now they're saying and so they can do that. They don't have an FDA that says we're going to just stick by the science.
they have an organization. I don't know exactly what it's called. And I put in a link to a PDF that group has put together. The public assessment report from the Medicines and Healthcare Products Regulatory Agency about using 1 dose of AstraZeneca instead of the AstraZeneca vaccine instead of using 2.
Do they have data on 1 dose only?
They have the data that I was looking at was neutralizing antibody data, and I was not impressed.
It doesn't sound like a good idea to me, but interesting.
Right. So for example, I'm looking right now at, and they actually do it with both the low dose or standard dose after the first one. And so here they show they have 6 weeks, 6 to 8 weeks, 9 to 11 weeks, or more than 12 weeks after the first dose. And so with standard dose, the patients had 52.8 as their GMT, or 52.98 as their GMT. After the first dose, 8,618.
You're talking about antibody titers, right?
Yes, this is antibody titers. So 8,618 after the first dose, 63,000 after the second dose.
Ooh.
Yeah.
Like you said, I mean, I had the same reaction as you. My recollection from the phase 2 trials, which looked at only antibody, was that in every case, the boost really had an effect. Now, as you say, we don't know what the correlates of protection are really, but the boosting is doing something.
Exactly. So I was really not terribly impressed.
But those are not efficacy data. Those are just antibody titers. I don't think because we went over the AstraZeneca paper, but they didn't have any efficacy after 1 dose, right?
I'm double checking right now. So they just have the data in terms of how many cases happened post until week 12. And they had 12 COVID cases out of 8,000 in the vaccine group and 44 out of 8,000 in the placebo group for a vaccine efficacy of 73%. The confidence interval is 48 to 85.
2 doses, right?
No, that's for 1 dose.
That's for 1 dose. Oh, okay.
That's table 10 on page 35.
All right, fine. But all right, so that's not great, but it's better than nothing, I guess.
I guess.
And so they've decided to do it. That's fine. We'll see what happens there. It's an interesting experiment. Okay. There you go. Now, last week, or maybe, sometime last week, there was a Times article that we discussed briefly with Daniel Griffin quoted as saying that the CT value is a predictor of clinical severity. And we were like, where are the data? And Daniel listened, and he sent us a paper that has some data on this in it. So he actually listens to every episode of TWiV, Dixon, and he's a busy guy. So I appreciate that. He actually said, here are the data for the role of CT values in clinical severity. He said, parenthetically, I'm really more interested in the relationship between CT and infectivity. That's what interests him. But these are the data out there, and this is the basis for his statements in the article. And there are more than one article, but this is one that I have picked, which is, what the hell is this? OFID, Open Forum Infectious Diseases. It's a brief report, and it's called SARS-CoV-2 Viral Load on Admission is Associated with 30-Day Mortality. And this is out of the University of Washington. And the Fred Hutchinson Cancer Center, and Aqualis LLC, all in Seattle. And the first author is Andrew Bryan. The last author is Alexander Greninger. And this is basically an observational study where they took. All right, so the question is there an association with viral load? And they also looked at antibody testing, yeah, and clinical severity. So they took existing patients and patient records to do this. And I just want to clarify something. In the first paragraph, they say, despite increasing evidence that viral load is associated with clinical outcomes, and they have 6 citations for that, and that high viral loads are associated with fewer tissue and culture infective viral particles. And there, when I read this, I'm like, am I stupid, or what are they saying? and I went to look at the 2 papers, and we think that they mean high CT values. That's what they're saying, right? Because high viral loads per se, independent of whatever you're using to measure it. When you say to me high viral load, there's a lot of RNA in whatever you're looking at, and that should have more. But the high CT value, of course, means less nucleic acid.
So that phrase has to be a mistake.
It should be high CT value, right?
Right.
Yeah,
they just got
that
tripped
would
up
make
in
sense.
higher CT means lower virus, lower CT means higher
But they do
virus.
it throughout the manuscript. It's not a one-off thing, right? So it's not higher.
I'm not sure that's consistent throughout the manuscript, but it doesn't.
Yeah, that's right.
Whatever.
Because I know.
Well,
go ahead.
What I was going to say is that's sort of the most egregious and glaring one. What they do throughout is talk about kind of like an association between virus load and the antibody response, and they don't ever say that it's an inverse association, because we talk about inverse correlations. So that was several times where I was inserting, things or circling it. But yeah.
Totally, yes, I agree with that. They tripped me up several times because they didn't say what the relationship was. They just said there was an association. And as we know, it can go in 2 different directions, right? So, in the AIDS HIV field, where patients are monitored frequently for viral load, that's more viral load, more viral RNA in your serum, right? It's quite clear. They don't get tripped up with the CT value. They understand that, a high CT is low viral load. But, okay, so I read this, and then I went to the show notes, and you guys had already picked up on this and said it was wrong. All right, so anyway, despite increasing evidence that viral load is associated with clinical outcomes and that high CT values are associated with fewer infectious viral particles, the FDA has not issued a EUA for quantitative assay. That's something that Daniel's been saying for a long time, right? He wants CT values. And this is part of the evidence why we should get them. So, and they also do serology assays in this as well to see if they can find a correlation. So what they have is 245 patients at UW medicine hospitals, and they have RT-PCR results and serum antibody results. And they have these patients in different age groups. They range from 10 to 20 years all the way up to 90 to 99 years of age. Wow. 99 years of age. That's great.
I'm just hoping I get that far.
Yeah. Yeah, I agree. That's amazing. I mean, wow. The primary clinical encounter for 194 patients is inpatient, right? And then there's emergency department for 39 and outpatient for 12. So that's how they were first seen in those kinds of situations. And some were asymptomatic at the first PCR result. And so then they look at 30-day mortality simply by manual chart review. They look at the records of these patients and the ones that had the PCR result or an IgG result from the day of admission. And they said, okay, what's the trend? If you're depending on your CT value or your antibody level, is there a trend in terms of your 30-day mortality? So they go through the data and their results. It's very short. It's like a 2-paragraph results section. There's more discussion than results, although the data, the charts are big. Okay, so first, interestingly, they had only one seropositive individual with a simultaneously CT of less than 22. So that's interesting. Less than 22, that's a lot of RNA, more RNA, less than 22 than, say, greater than 30. And yet they have only one seropositive person with a low CT value there. So they say IgG values as measured by index values were found to be inversely correlated with SARS-CoV-2 viral load. So the higher your IgG, the lower your viral load. Is that correct? Mm-hmm. Are they using viral load correctly there?
I believe so.
They're using RNA as their surrogate for viral load. They're not measuring infectious virus, but right.
I just wonder if they're using CT values, which...
Yes, I think that's the basis of in this graph in Figure 1 where they've got the CT values on the y-axis and the immunoglobulin data on the x-axis. So that's what, and that one red square in the bottom right quadrant is the one that they're talking about.
Yeah. Okay. Then to test whether seroconversion or high viral loads were associated with mortality, they examined the clinical data, as we said, and they conclude that the viral load on admission was found to be independently associated with mortality after adjusting for serology, age, and sex. So if you had a high viral load on admission, and they define that as a CT of less than 22, they had a significantly greater odds of dying. So there they got the viral load right. High viral load, CT less than 22, right?
Yeah.
So your conversion trended toward lower mortality, but they say that they didn't have enough data to be statistically significant. That's it. That's the finding, which is saying, okay, if you have a low CT on admission, you maybe going to have a more difficult clinical course.
So I ain't no doctor, but I assume that the utility of these sorts of data are that, it's, I guess, sort of prognostic in a way, so that it might influence how you approach treatment.
Yeah.
To individuals, depending on their virus load coming in.
Right. I think that was the gist of the quotation that I pointed to in last week's article, that Daniel was saying this could be useful information. And true confessions, when Vincent said, where's the data, I was looking at my hard copy of it. When I went to look at the online copy, the journalist had put in 5 or 6 references to other papers, 3 of which are published and 2 of which are preprints. And just briefly mentioning that there are some of them that have even higher sample size, up to over 1,100 from Mount Sinai, and that's in the Lancet. There's a Nature paper. There's an Annals of the American Thoracic Society. So those are all published. And then just to say, how global this is. There's a preprint from Sao Paulo, one from Madrid, and one from Bahrain. So, if somebody did a meta-analysis on this, they could probably get even more powerful data. But basically, they all at least find this correlation between the CT values and the outcome with respect to death. And then some of them look at other outcomes and see some kind of correlation, but that might not be statistically significant.
So one thing that I find somewhat interesting looking at the data, I'm specifically looking at panel I in their figure 1. And Kathy, I didn't look at the other papers you have mentioned, so I don't know if this is similar. They talk here about the cutoff of 22 as their CT. And, I would, caution people against making a big difference, making a big deal about, this patient has a CT of 21.9 and this patient has a CT of 22.1. And so there's going to be a huge, huge change. I don't know if the other papers also settled on CT of 22, but it is sort of interesting in figure I. It almost looks like there's a little bit of a gap. There's sort of patients clustered at different CT levels, and there are relatively few that are sort of between 21 and 23 or so. So I think that is sort of an interesting, obvious place for me to draw the line, but I don't know why there's a gap
so
there.
To me, it reminds me of when you do flow cytometry and you decide where do you put your gates. That maybe they looked at this and put their gate there. I also don't know what the other papers used in terms of a value, but I was going to say something else and now I forgot.
But they do say in the discussion, so our data suggests that CT of 22 may be a useful cutoff. However, sample and swab variability across patients may limit the widespread use of a discrete cutoff. So you can't say 22 everywhere.
Right. But I think from this paper, they're also, based on this, what they show in figure 1A, where there's only that one red square patient that surprisingly had high virus load and had a measurable IgG response, that if you use those two things together, and the IgG response is going to come from blood. So you don't just use one thing. You don't just use the CT value. You use the IgG data as well. And together, that could give you a better prognostic indicator.
So the upshot is that I think Daniel has said that the FDA is going to authorize release of CT, right, for those assays that measure it, which is good.
Well, in fact, in that article last week, 2 FDA people speaking off the record said there was no limitation to giving that data in the first place. But
Daniel always said that it was never authorized, right? He would say, they're not authorized to give it, but so now we can get it. So that's good. And as Daniel said, so you can use it as a indicator of clinical severity, but in his case, he's interested more in, if you come in and you have a CT of 37, you're either at the end of an infection or at the beginning of an infection. And even Michael Minna talks about that. If you have, in the past, if you had a CT of 37, you were positive and you had a quarantine. Well, maybe there are a lot of people, I think in his editorial, he said like 50% of the assays give a high CT and they're probably not need to be quarantined. So we can use that information to determine quarantine as well. And that's important, I think. Okay, so there you go. That's that. Thank you, Daniel. The other manuscript I noticed, and it has to do with, all right, so one more thing I wanted to mention about this study. So if you have a high viral load, a lot of RNA copies at admission in this study, for example, you're more likely to have a serious clinical outcome. I just want to point out that does not mean that you were infected with more virus particles. All right. It just means that at the time point taken, you have a high viral load, and that's because the virus is reproducing better. And why that is really the question for those people that go on to a poor clinical outcome. We don't know. It has nothing to do, as far as we know, of inoculum, because this is another issue that we've visited for a long time, this idea that inoculum determines severity of disease. But this paper doesn't address that. However, the second one does a dose-response experiment in ferrets, and I thought it would be interesting to look at that and get some information from that. Now, ferrets are not people, as we know, and
I know some people that are kind of like ferrets.
So mice lie and monkeys exaggerate, and what do we say about ferrets?
Ferrets are not humans.
Just, okay.
Yeah, and there's a button on Cafe Press that says that, and one of our listeners made a work of art with that on. Yeah, I mean, if you think about it, the ferrets interact differently from people, and although, as Rich said, some people have properties of ferrets. Their respiratory tracts are smaller and so forth, so you can't extrapolate. But I think the study is interesting to look at. It's published in Nature Communications, dose-dependent response to infection with SARS-CoV-2 in the ferret model and evidence of protective immunity. And there are actually a lot of data here that we have touched on before in other ferret studies, but the dose response is what I wanted to look at here. And Rich, you knew somebody on this paper, is that right?
Yeah, the corresponding author, the last
Miles
author
Carroll?
is Miles Carroll. He was, I know him from Pox days. He studied with Bernie Moss, I believe, as a postdoc, did a lot of work on vaccine vectors and, Pox recombinant vectors, MVA, et cetera, and went off to, after that, work for a company called Oxford Biomedica. He's British by origin. Worked for them for several years. And in 2008, let me make sure I get this exactly right. 2008, he took over as Director of Research at Porton Down. And says one of his responsibilities to the UK government is the rapid development of in vivo models to assess pathogenicity, transmission, and medical countermeasures for outbreak pathogens. And so for those who don't know, Porton Down is one of the sites of Public Health England, and it has high containment facilities, et cetera, where they can do research such as this. So
always liked it.
Yeah, Director of Research at Porton Down is a position of significant responsibility.
I always liked that name, Porton Down. It's great.
Mm-hmm.
I also like Miles as a first name, too. That's pretty cool, Miles. I like that. So this comes out of the UK and also the Doherty Institute in Melbourne, Australia, and also Oxford University. First author is Catherine Ryan. Oh, look, the 15th author is Karen Buttigieg. I wonder if she's related to Pete. Just kidding.
And if she pronounces it Buttigieg. Like
Buttigieg, or whatever, yes.
Yeah.
Okay, so we've known for some time that ferrets can be infected with SARS-CoV-2, and they develop typically mild disease, and they can be used to study some aspects of the pathogenesis. But what I thought was interesting here is they infect the ferrets with 3 different doses of SARS-CoV-2. So they're infected intranasally. And here I got thrown by nomenclature, which I guess is different in the UK, but they call their infection challenging, right? Mm-hmm. I typically reserve challenging for, you immunize and then you challenge, but they're calling it all challenging. Ferrets were challenged intranasally in 1 ml volume to increase the incidence of virus reaching the lung. Interesting. At 3 different titers representing a high, medium, and low dose. Okay, 3 titers. And so you can see what happens in the ferrets at the 3 different titers. And then they look at shedding, and they do a very extensive shedding study from nasal wash, throat swabs. They go down to the lung, bronchoalveolar lavage, nasal cavity, tonsil, trachea. Wow. So it's very extensive. And go ahead.
I just want to comment on what the doses actually are. The low dose is 500 PFU per mil, and they're delivering, I believe, a mil. Okay, they say in half mil hits. I think that's correct. So the one paper that I've seen that has and I'm not comprehensive at all, that has any indication as to what an infectious dose of SARS might be in humans relates to SARS-CoV-1. And this is a very hard number to get. They came out with ballpark 250 particles, 250 infectious units, I think, as an infectious dose. So 500 in a ferret is at least within an order of magnitude around the same as that. That was their low dose. The medium dose was 100 times more than that, 50,000. And the high dose was 100 times more than that, 5 million. So it's a broad range, and the high dose is a lot of virus.
And what this means for people we don't know, right?
Right.
Because people are not ferrets. So, yeah, and I mean,
Right.
remember, this is being pipetted right into the nose, right? They're anesthetizing the animals so they're not moving around. You pipette it in, and I don't know how they're doing it, but, you know, when we do it in mice, we kind of tilt them to make sure it stays in the nose, right? It's not what happens in people, so you can't
Well, I don't
extrapolate.
know. I have some bar tricks that I've seen. I mean, belly shots. How far removed is that from this? I don't know.
So what you're saying is some people can behave like ferrets, right? Anyway, and now see, all of my highlighting is gone. I highlighted this last night, and for some reason it's all gone. So we'll have to search. So, all right, let's look at nasal wash, viral RNA by RT-PCR. In the high dose group, 6 out of 6 ferrets, they found RNA from day 1. And again, it says PC, and I'm like, oh, is that politically correct? Oh, it's post-challenge. Day 1 post-challenge, and they could find it until day 20, peak of shedding between 2 and 4 days in the high dose group. And then the RNA goes down, and then all of a sudden at day 16 and 18 it goes up again. I don't know what to make of that. Please don't say it's integration and transcription. Please. I don't think that's what it is. And then they got infectious virus in the nasal wash of 2 out of 6 of the high-dose ferrets at 3 and 4 days post-challenge. And so it's not easy to get infectious virus out. And they say maybe it'd be an issue of the assay because they're centrifuging out the cells, and that may remove the virus. For the RT-PCR assay. In the medium group, in the medium dose, 6 out of 6 also had detectable viral RNA from day 1 post-challenge. The peak was a little more variable, and then eventually fell below the limit of quantification. Nothing detected after day 16 in terms of RNA. No infectious virus in the nasal wash of the medium and low group. So only when you give that highest dose, 5 times 10 to the 6 PFU, right, because they're giving them a mil, do you see infectious virus? Quite interesting. But obviously something's going on there because you're getting viral RNA. And they're looking for subgenomic RNA, so it has to be reproducing somewhere. And we'll see that there is evidence for that too. Where's the low group? I think they also saw, I've lost the low group. Does anyone know where it is?
The low is always the green.
I'm looking at the text.
Okay.
I'm a text kind
the
of
low
person.
group is about 4 lines up. Sorry.
Peak of viral RNA, decline, blah, blah, blah. Quantifiable. Oh, here we go. RNA was only found in the nasal wash of 1 of 6 ferrets in the low challenge group. Right. Because you got 6 out of 6 in the high and medium. Low, only 1 of 6. So really hard to get establishment at 500 PFU. I
just finally
in a ferret
figured out what subgenomic RNA means. So I presume that is an amplification that includes primers for both the leader region and the coding region of a subgenomic RNA. So it detects specifically messenger RNA for structural proteins and stuff like that. So that's a very precise indicator of there is virus replicating here.
Yep.
Okay.
Right.
Sure.
They don't say that till the very end, but yeah, the subgenomic RNA is the measure of replicating virus in their hands.
All right. So they do a lot more quantification in other ways, but that's good enough. Unless anyone wants to bring up trachea and rectal swabs, that's the point, that there's this dose dependence. If you don't put enough virus in, you don't establish an infection. Presumably it's cleared. Like the low dose is just not enough in a ferret to establish an infection. So then clinical signs, does it differ? let's take a look at that. At day 9 post-challenge, all 3 out of 3 ferrets in the high dose group showed reduced activity. They don't move around as much. They have ruffled fur.
Yeah, those are their 2 clinical indicators. How much activity is there, and whether or not they have ruffled fur.
Right.
That's
So
it.
the ferret. Go ahead.
I was just going to say they use the word playful. They're not as playful.
Oh, that's very interesting. Yeah, and ruffled fur, ferrets groom themselves, and if they don't feel like grooming, they're not feeling good, so they don't groom themselves, so their fur is ruffled. So they're going to stay home that day because they're not.
Maybe that's one of the cases where ferrets are like people.
Yes. They do look at things like temperature and weight loss as well for their clinical data.
So there's a little bit. So there's no weight loss. The weight loss is a nice way of measuring illness, and they say there's no weight loss, but in fact, the ferrets failed to gain as much weight as the ferrets in the control group. So over the time of the experiments, they'll gain some weight, I guess. All right, so that's the high dose group. The medium dose group. So they give a clinical score, right? And it's 14 in the high dose. And then the medium dose group has 6.99. So same signs, but less frequent. And low dose, sporadic instances. They don't even come up with a number for that. And that makes sense because only one was shedding RNA. Then histopathology. They euthanize some of these animals, and they can look at the nasal cavity. They can look down into the lungs. And they say in the nasal cavity, you see a minimal to mild necrosis of cells, mild inflammation, consistent with infection. And they can stain the cells for viral RNA so they can see that individual cells are infected. No remarkable gross lesions observed in the animals. If they look at the lungs of the high and medium dose groups, they can't look at the low because only one was shedding RNA, so there's nothing there. But in the high and medium dose groups, a mild multifocal bronchopneumonia was observed. All right, which is associated with infiltration of inflammatory cells. And here they have a little quantification. In 10 to 15% of the lung sections at day 3, they see infiltration of inflammatory cells, which they can say are mostly neutrophils, macrophages, and lymphocytes. In the medium dose group, they see the same infiltration in less than 5% of the lung sections. So that's actually a key point in my mind, that here you have the high dose group, you have more lung sections showing some kind of pathology. It's a mild pathology, but it's there. It's obviously there. And if you put a lower dose in the medium dose, you have less. So there are less foci of virus reproduction, basically. So, and then in the low, of course, you don't see anything. So does that translate into, what is the claim that people make? Higher dose of inoculum, more severe disease in people, right? So I suppose if, we can't say that this extrapolates to people, but it's just ferrets. If more of your lung is compromised because you have a higher inoculum and more extensive reproduction and therefore more damage to the lung, you're going to have a more severe disease, I suppose. Is that reasonable thinking?
Well, certainly that's a logical train of thought. Okay.
Yeah.
Whether that pans out scientifically or not, I don't know. But that's
Right.
We don't know.
Right.
In people.
Whether that's the mechanism. Yeah.
Right.
Yeah, we just don't know.
I do sort of find it interesting that they actually are not showing the low dose here, because I think that there are lots of people who are making an argument about, well, if you're just exposed to a very low dose, it's fine, or it could immunize you or things like that. And so I would actually be interested to see some of these data with the low dose animals.
Yeah, just take out lungs and see if there's anything there at all.
Yeah, exactly.
Right? Yeah, I agree. So you can see here with a very low dose, very little happens, right?
Right. But then we don't see, we don't see PCR RNA in the nose, but does that mean we don't see RNA everywhere?
Yeah, I don't know the answer to that. Yeah, but I think one of the reasons I wanted to discuss this paper is because many people are emailing and saying, "Oh, there are all kinds of animal experiments showing dose-dependent disease," and I said, "Really? I don't know about them." And I went back at the Barrick papers and the non-human primate papers. Remember Emmy DeWitt? We talked with her a while ago. They only use one high dose because they can't afford to do different doses. Here with the ferrets, they could do 3 different doses. They're not as expensive as a non-human primate, but you can't do a dose titration, 10 different doses. Forget it. It's too expensive. And I haven't seen anything. This is the first paper where I've seen 3 doses and following what's going on, not just in shedding, but in pathology. So in my view, there's no evidence that, except for this in ferrets, that, more high. In fact, they say they all had a mild disease, right? So even though you look in the lung and you find more sections with pathology, it doesn't translate to a higher disease. That's the point I'm trying to make, I guess. This is the only animal experiment.
One of the things that interests me about this, they have 2 animals that they call naive sentinel animals. And I can't figure out from the methods how exactly a sentinel animal is treated, whether they're in an adjacent cage or in the cage mixed up with the animals that were actually inoculated. So I'm not sure of the details about that, but I presume that somehow or other they are exposed to the animals who have been inoculated with virus. And at the end of the results here, towards the end of the results, they say they have just 2 of these sentinel animals. They say 1 naive sentinel group ferret was shown to have neutralizing antibodies to SARS-CoV-2, as well as the presence of SARS-CoV-2 spike-specific IgG upon euthanasia at 20 days post-challenge. No clinical signs in that animal. No evidence of pathology. Importantly, to me, that's evidence of transmission. Okay, very small numbers, but it is apparent, and they don't conclude transmission from that. They just point out the observation, which I think is a wise way to present this. But my interpretation is that that's at least some evidence for transmission in this particular model,
which has been published by others in the previously by putting ferrets in the same cage, I believe.
Mm-hmm. Yep.
So here in the discussion, this is the one sentence I wanted to highlight. Both doses, high and medium, also induce classical pathology of bronchial pneumonia involving 10 and 3% of recipient lungs. So it's the same disease in both cases. Bronchial pneumonia is just more extensive at the higher dose, which makes perfect sense, right? And again, whether that happens in people, we have no idea. We're not likely to ever know because you cannot do this experiment in people. And I don't know how else you would know the inoculum that people get. So everything else is speculative in people anyway, So, okay. That's all I wanted to do on this. Does anyone want to bring up anything else?
I mean, they do have a little bit of data here about rechallenge of some of these ferrets.
They do. Yeah.
Trying to indicate whether, having a particular dose for your first infection will influence what type of immune response you make at second infection. I don't know that I have a great take-home message from those data. But I think
pretty small N.
It's a very small N, and there's some noise in the data. So I will say that that data exists here, or those data exist here, though I'm not sure that we can make huge conclusions from it. But we certainly can't. Again, we certainly can't say that, the low dose was amazingly protective or the low dose was not protective.
Yeah, I agree. Okay.
The one other thing I would say is that Miles pointed out to me that they've done experiments in both rhesus and cynomolgus macaques that are in review. It's been published on bioRxiv. You can find that there. That's not a dosing study, but it is a study of the pathology in a non-human, in 2 different non-human primates. So people can have a look at that if they want.
Okay. Let's do some email. Rebecca writes, I hope you are all well. Leading up to the Christmas period, it's traditional to send Christmas cards to friends and relatives via the postal service in the UK. It occurred to me the other day I'd received at least a dozen Christmas cards, all with envelopes sealed shut. This is usually via licking the sticky part of the envelope. I've seen some literature suggesting the persistence of SARS-CoV-2 on paper could be as long as 21 days at 20 degrees C. Wondering what your thoughts might be as to whether this traditional activity would have contributed to the spike in cases, 30,000 plus per day now in the UK, despite the tiered restrictions being in place. Kind regards, Rebecca is a lead microbiologist at Baxter Healthcare UK. Well, I guess most people open an envelope by putting their finger in a little crevice, right? And that ends up rubbing your finger against the glue, right? Very few people use a letter opener. I mean, I have a letter opener, but I never use it. I don't know why.
I always use a letter opener.
Do you?
I always use a letter opener. There's my letter opener. I actually
it makes
have 2 of them.
perfect sense. They're much easier than putting your... I'm such a jerk. I'll be sitting there struggling one end of the envelope, trying to get a finger, and then I go to the other end, and here I got this thing sitting on my desk. I'm such a ferret, right? I think this is probably a low-risk activity. I doubt very much it would have contributed to the spike in cases, but I have no scientific data on which to base that on. Right?
Also, I went back to find the paper that I could easily find about the persistence of viral RNA as measured by PCR on paper at room temperature, 3 hours to 5 days. So, but that's just the RNA. It's not infectious virus.
Yeah, in 5 days there might not be enough virus. Yeah.
Also, I would add that, use a letter opener if you're worried. Wash your hands after you open your mail. And probably, based on my experience, fewer and fewer Christmas cards are being sent every year. And I don't think it could be enough to explain a large increase in cases.
Yeah, I would agree. And I've stopped sending cards years ago, so it doesn't. But I do get envelopes of other kinds, right? And I guess some of them are licked. There is obviously virus in saliva because some diagnostic tests use it. And it's probably quite a bit. But it's a good idea, but I think it's a low-risk activity. Personally, I'm not worried about the mail. No, I'm not worried. We were at the beginning. We were a little cautious, but it turns out that it doesn't seem to be a high-risk activity. So,
I go walking around my neighborhood, and there's gangs of kids out playing basketball and other sorts of activities that, even though they're outside, they're not masked. They're in close proximity to each other, and it just makes me cringe. Okay, because that's, to me, that's they're pangolins, right? That's the intermediate host. Okay,
pangolins.
Did you hear that Boston University and I think it was Holy Cross played a basketball game, all masked? They all do their practices masked. And the coaches both said, yeah, we can do this. It's a little bit different, but we can do it. And so it may have been the first NCAA game where that was reported or done or something. I don't know. So
I haven't heard that. I'll have to look into it.
Didn't know. Brienne, can you take the next one?
Sure. Shoshone writes, hello, folks at TWiV, and greetings from Omaha, Nebraska, where it is unseasonably warm today with expected high of 58 Fahrenheit and sunny. I apologize if this email makes no sense, but trying to explain my question is difficult. Here it goes. I have now run across 2 people in my life who are choosing not to get the vaccine because of fertility concerns. Both are citing statements by a Michael Yeadon stating that the spike protein and a placental protein, syncytin-1, share a protein sequence. The theory he is sharing is that the vaccine produces antibodies against the spike protein and would thus also produce antibodies that, in quotes, may attack the syncytin-1 protein, leading to failure of placenta formation. I see multiple issues with the statement, but my biggest question is one of logic. If you get a natural SARS-CoV-2 infection, won't you also then have antibodies against syncytin-1? Therefore, couldn't a natural SARS-CoV-2 infection cause sterility, along with increasing your risk of death, permanent lung damage, heart damage, kidney damage, stroke, etc.? Thus, the risk of a natural infection far outweighs the risk of the vaccine, right? In addition, I read that syncytin is one of those proteins found in abundance in ovarian and endometrial cancers as well. So by his logic, the vaccine may have anti-cancer properties. Just kidding. Definitely not suggesting this is a thing. Thank you for all of your hard work, humor, and dissemination of the nerdy science information. Your podcasts are one of the highlights of my week. I have been able to use what I have learned from your podcast in both my professional, EM physician, and personal life to help people who feel lost and hopeless. Sincerely, Shoshone. So I agree with your logic that if we take these syncytin data at this value, then yes, natural infection would be have this additional risk. I haven't looked at the data on syncytin in a while, but I think when I last looked at it, the similarity that was discussed here was a sequence of 4 amino acids in a row between syncytin-1 and the spike protein. And the way that I described that to a non-science friend was noting that it is probably likely that if you looked through all of the things I've written in my life and all of the things that Shakespeare wrote in his life, you would find a place where we use the same 4 words in a row. But that does not mean I am in any way, shape, or form Shakespeare. And this is not going to be enough of an overlap or enough similarity in amino acids to really lead to an antibody response that would be cross-reactive.
And what are the typical sizes of epitopes anyway, right? More than 4 amino acids?
More than 4 amino acids. It's a little bit harder in the case of an antibody response. It's more straightforward in a T-cell response, but it's, I'll ballpark and get yelled at by immunologists and say 8 to 10 is a minimum.
Okay, that's what I would have guessed.
Shoshone, you get my critical thinking award for the week. Really very nicely done. Okay, and boy, these people who come up with this stuff, what are they doing? Like this Mike, I'm talking about Michael Yeadon, the guy with this idea. It's nuts.
Do we know who this is?
We don't,
no.
but I have come across this myself pretty frequently in terms of friends asking me this very question.
Yeah, I'm glad you were prepared with the amino acid sequence homology because this is the first I've heard of this.
Yes.
Well, he knows some science. He's an allergy and respiratory therapeutic area expert. Because I was going to say, like you were saying at the top, Rich, if you're not familiar with the process of science, then you can get, but he may be. I like, Brianne, that you said identity, not homology. It was really good.
I was being very, very careful.
Yeah, you paused. I could tell. Yeah, what's the right word now? Yeah, very good. Otherwise, then we get more people yelling at us, right? Yeah, this I don't think there's been any association with COVID-19 and sterility, right?
I have not seen any, no.
Not likely. Kathy, can you take the next one?
Yes, Swan writes, "Hi guys, listener from the Middle East. Thanks for all the good work. What do we know about the Sinopharm vaccine results? It has been approved in the UAE, United Arab Emirates, and they are offering it to any resident. I can go get it, but the results from stage 3 haven't been released. Would you guys recommend me to get it? Best Juan. So I had looked ahead last week and put in links of what we knew up to that point, but the day that we recorded on December 30th, I heard on the news that the Sinopharm results were announced. Now, I haven't gone to find an official announcement of those results, like a press release from Sinopharm or something else. I only found what I had last week, which was this BBC article that showed that the phase 3 trials said that it was 79% effective. But UAE claimed that it was 86% effective, but that was based on interim results. So the final results, at least according to this article, were 79% effective. We discussed the fact that it's an inactivated virus vaccine, I believe, and I'm not that kind of doctor, but a vaccine, if it's available and 79% effective, and if they have the appropriate safety parameters, consider going ahead and getting it.
Yeah, if it's an activated vaccine is old tech, it should be okay, right? Mm-hmm. In fact, someone asked me the other day, why didn't any U.S. company develop an inactivated vaccine, right? And I don't know the answer. I don't know what they're thinking. It's old tech. Maybe they wanted to use new tech, which would be easier. And maybe they didn't want to grow up hundreds of liters, thousands of liters of SARS-CoV-2, right, which is a containment issue. But obviously they did it in China, so I don't know the answer. I just said if Jonas Salk was alive today, he would have done it because that was his shtick. So we'll set to see what the data are here. They haven't published them, obviously. I don't know if they will.
Yeah, the safety risk of growing up large quantities of a virus for which there is no existing therapy or vaccine, could be a major reason not to approach that first.
You would have to grow it up under BSL-3 conditions, I presume.
I would think so.
Absolutely.
And it's also going to be a technical challenge. Mm-hmm. And there's also, with inactivated vaccines, there's the issue of identifying an appropriate method of inactivating that inactivates the virus without compromising its effectiveness as a vaccine. Yeah. So this can be tricky.
Yep.
Right. And then one thing that I think that was known from some of the work with MERS and some of the other coronaviruses was the way to make this post-fusion or stabilized pre-fusion form of spike.
Yeah.
And so that wouldn't be available with an inactivated vaccine, and so if we knew that there was sort of this optimal form of spike, it makes sense to go for that instead of using perhaps what might be a less optimal version that's in the inactivated vaccine.
Rich, can you take the next one?
Elizabeth writes, Dear TWiV team, first off, I agree with pretty much every positive thing I've heard your other listeners say or write in. Thanks so much for doing a show. Just writing today because I saw a slightly macabre joke on Twitter I thought you might like. I'll translate the German for you. The people from whom you really don't want to hear the words, this does not look good. Oh, okay. Here are the people from whom you really don't want to hear the words, this does not look good. The plumber, the mechanic, the hairdresser, the dentist, or Christian Drosten. Very good. Stay safe, Elizabeth. Oh, P.S. Maybe I should credit the tweeter. Their handle, if that's the right term, is at Dr. Wow Meow. That's W-A-U-M-I-A-U. If I were to read it aloud, it would sound like Dr. Wow Meow. That's the German. Yep.
So what would a hairdresser say that does not look good?
If they cut your hair or dyed it badly.
And they cut it
turned out
all wrong.
The color was bad. Yeah.
But you're looking in the mirror, though. That's not like a dentist where you can't see anything, right? But I agree.
It's not fun when it happens.
No, it's happened to you, Brianne?
Yes, it has happened to me.
I have the picture of the operating room where the surgeon says, "Uh-oh."
Yes. Uh-oh.
Oh, I have the picture up in my lab of like somebody in a glove box or some one of those. I think it's a Sidney Harris cartoon where they say "uh-oh" and the flask is broken or whatever.
Yeah, that's right. That's a good one. Yeah. Right. Brenda writes, "Dear TWiVers all, thank you so much for the plain speaking about what is going on in the UK in your latest podcast. I have recommended your podcast to as many Scottish MPs and MSPs as I think will listen. I have no belief whatsoever that this variant is more infectious. I live in Scotland, where virus prevalence has been low and falling. Why? We have our own trace system with trace rates in the 90s. The Scottish trace system is the devolved Holyrood government in an NHS of Scotland. We are partly dependent on the UK Lighthouse test system, but ScotGov is working to reduce our usage. From bitter experience, we know they do not function properly and must be monitored closely at all times. The problem is the test and trace system in England sucks. I would suggest calling it the test and failed to trace system, since the latter description is pretty much what Deloitte and their subcontractors implemented. It was designed as a totally centralized private system and did not involve local English NHS or public health professionals. It is still failing. For example, just the other week, they failed to notify the Welsh authorities of 1,100 positive results due, they claim, to scheduled system maintenance. As a retired IT professional, I am incandescent at such a lame excuse. I got to remember that word, incandescent. That's good. Last I heard, and it is impossible to find out the figures, the trace rate in England was in the low 60s and struggling. I know it is not strictly virology, but do you know yourselves, or do you have any statisticians or public health contacts who can tell you what the effect of such a poor trace rate does to infection rates? I think we know that. We've been told, right? Many people. Brenda's in Black Isle, Scotland. P.S. WHO by nature as a multinational body has to be more circumspect. Watch the last two press conferences. For every question on the new, quote, strain, watch them not say it is more infectious, and then go on to experts stress the use of NPIs and having a functioning test and trace system. They have been banging on about this from the get-go. Thank goodness you are not bound by the restrictions that prevent them from speaking some home truths. Yeah, we are not bound by anything, really, except science. That's what we are bound by, to give you the good science. Anyway, if you don't test and trace properly, it's a big problem, right? It increases infection rates. But on the other hand, if infection rates are so high, it's very hard to implement because so many people are infected. You just can't do it because you're limited by the people that are needed to do that. There's the, is that
I was wrong. It's a Far Side.
Far Side, right.
Center for the Study of Viral Pathology, and they're looking out on the sidewalk at the dropped flask from the window 3 stories up. Yeah.
Isn't that funny how everyone's walking around in lab coats?
Yeah.
Yeah.
Well, it's the Far Side world.
It's a campus.
Yeah.
People are, anyway. Okay. So the next one is not for reading, but it's for you to enjoy. I hope you enjoy. I don't want to provoke anyone. So, Brienne, can you go to the next one?
Sure. Terry writes, "Hi, Vincent and the whole TWiV team. I am another fan addicted to the knowledge that you each kindly and intelligently provide every week. As many others have said before me, I do not know how I would have survived this pandemic without you keeping me grounded in science and educating me on virology. I have learned so much and enjoyed every minute of it. I am just an HR manager in a large entertainment company, but I was born with an insatiable curiosity and a love of science. In recent years, I became fascinated with neuroscience, then the microbiome, sleep science, and now, of course, virology. So as the citizens of this beautiful blue marble of ours prepare for our newest miracle of science, the COVID-19 vaccination, it is in all our best interest that these vaccinations are as effective as possible. I have read that getting a good night's sleep, both the night before and the night after vaccination, can increase the vaccination's effectiveness. Can you please comment? And if this is backed by science, please encourage your listeners to prioritize their sleep window when they receive their golden ticket. Thanks again for all you do. And Terry is from Orlando, Florida, where it is currently 54 Fahrenheit and clear.
Golden ticket, huh?
Yeah, and I think I've referred to it as that, I think, or at least similarly.
Isn't that from a movie where you get a golden ticket and you get a chocolate bar in the chocolate bar or something, and you get to go see Charlie the chocolate guy or something?
Yeah.
You get to go see Willy Wonka, yes.
Willy Wonka.
Okay. Brian, if you get a good night's sleep, will you make a better immune response?
There is some evidence, though it's not very strong, that sort of improving one's general health with good sleep routinely and good nutrition is positive for your immune system. I don't know if a one-off night of good sleep is going to make a huge difference, but it is generally thought that your immune system is a little better if you are in good general health.
Yeah, I agree. Now, one night before is not going to make any difference unless you're taking an exam the next day. Then you should get a good night's sleep, right?
Yes.
Especially Brianne's exam. You better get a good night's sleep.
I recommend a good night's sleep every night.
Yeah, it's a good idea. Not always easy to achieve, though. Kathy put another cartoon in. It's just hilarious. This is just so funny.
Yeah, this is another one I have up in my lab, so I'll describe it. It's two guys sitting in glove boxes, but the problem is that they're each on the other side from each other of the window. So one guy's stuff is in the same compartment as the other investigator, and his experiment is in the compartment of the first guy. So, yeah.
Hilarious.
Still not a Sidney Harris cartoon, but yeah, it's good.
Cool. Kathy, can you take the next one?
Stephen writes, Dear Team TWiV, I've been listening to your podcast for several months trying to improve my understanding of what is happening with COVID-19. I do not have a scientific background, but nonetheless, I found the material accessible most of the time and immensely enlightening. Thank you for all the great work. In TWiV 696, you discussed this paper on excess mortality for 25 to 44-year-olds, and he gives the link to the JAMA paper. I'm fully aligned with your view that, quote, even 25 to 44-year-olds should not be dismissive about the risks associated with the virus. However, I worry that your strong statements about the conclusions of the paper could actually risk pushing some people in this age bracket to believe that you are over-egging the pudding, as it were, and could be counterproductive. Let me explain. The paper, similar to other reviews and reports of this nature, provides no information regarding the circumstances or factors for the 4,535 COVID-19 deaths recorded. This may well be due to a lack of data. It leaves open the question, did these 4,500 individuals have underlying health conditions or circumstances that place them at unusually high levels of risk of death due to COVID-19? Without clarification of this nuance, then, for example, a 30-year-old with no known health problems who eats well, exercises, and generally takes good care of themselves could easily conclude that COVID-19 represents an insignificant risk to them, and that the 4,500 deaths in the group related to people with existing health problems and/or the 4,500 deaths are in the group related to people with existing health problems and/or who lead unhealthy lifestyles. To make a more general point, there seems to be a lack of data regarding the relative risk factors for COVID-19, whether in terms of death or serious illness or long COVID, across different population segments. Data for broad groups, such as 25 to 44-year-olds in this paper, gets reported, but there's a lack of any detail beyond the high-level segmentation, age in this case. Without that detail, people who wish to see themselves as low risk can easily conclude, well, I'm different from those people, and even worse, they might feel that the risk levels are actually being exaggerated. My questions to you are then, have you considered the issues raised above, and is my concern regarding the risk of counterproductivity valid? 2, is there data that can be used to help provide a more granular view on relative risks for different population segments? And 3, can and importantly should scientists, doctors, governments strive to deliver a more granular, detailed message about risk factors to communities? or perhaps they shouldn't. I would love to hear you discuss these questions in a forthcoming episode. Kind regards and seasonal best from Cheshire, England, Stephen. So I was not on the episode when you did that excess deaths paper.
Well, yeah, I mean, this that's actually co-authored by the next head of the CDC, whose name is not on the tip of my tongue, but she's an infectious disease doc, and the idea was, so many people have confirmed deaths in this age group from COVID, but in this year there are excess deaths, which you don't see in previous years, and they're likely attributed to COVID-19. But the death certificate did not say death by COVID-19, and we had another email about that as well. And this one suggests, well, maybe they have underlying health conditions. I don't think it matters. They're still in the 25 to 44-year-old range, and they may not even known they had. So it could be, yes, that they had health issues that they didn't know about and that predisposed them to death. But then, in the general population, a 30-year-old with no known health problems, that's the key. You may have them but not know about them. So that's why you need to avoid being infected, even if you sleep well every night, because you never know if you have a health problem that could lead to it, if in fact that explains them. That's my take on this anyway.
Does that address the questions? Yeah. Go ahead.
The future CDC head, I believe, is Rochelle Walensky.
Thank you.
Do I have that correct?
Yeah. Yeah.
So I think the one thing I would say that in addressing these questions is that there is sort of a worry in terms of making your data quite granular, as far as things like, at what point do you actually start making it possible to figure out which individual is which in your data set? If at some point when you get too granular, you're actually able to start to identify individuals, and you get some HIPAA issues or other types of ethics issues. I think that the take-home message of that paper, as it was written, and I think as we talked about it, was that there is risk in the 25 to 44-year-old age group, and perhaps it is unappreciated by some members of that age group. And so I am sure that there probably are 30-year-olds with no known health conditions who underappreciate their risk and say, oh, I have no risk. And I would just say that I don't think that there is. I think that this paper tells them that maybe they're underestimating that, and that's really what we can take home from that paper.
This concept of excess mortality, we do it all the time for influenza, because influenza is not always identified as the cause of death on the death certificate. Sometimes it's just pneumonia. So we look at excess pneumonia deaths in an influenza season as a gauge for influenza-induced death, and that's perfectly acceptable. And those people may have underlying health conditions. The point is we can't get any more granular because the death certificate doesn't tell you anything more than that. But I think at the end of this pandemic, we'll be able to go back and look exactly at how many excess deaths were associated with the pandemic. Next year, if there's very little activity, we'll see that go back down again. So I don't know that – I understand that you don't – what did they call it? Too many eggs in the pudding?
Over-egging
the pudding. My gosh, we're trying to inform you about your risk and so that you conduct yourselves safely. And I don't think that's an issue. But I have I have a different view. As I said somewhere else, and I can't remember where, after we discussed this paper, our 26-year-old came to visit, and he's very depressed that he can't lead his normal life, right? I mean, I saw him get very upset, which I'd never seen anyone else do before, even though they've written about it. And I understand what is motivating younger people to want to be with their friends. He's good. He doesn't do it because he understands the dangers. but he's suffering as a consequence, and many other people are. So I get it. I really understand it now. But I just think it's not a risk worth taking. Well, it's a risk you got to decide for yourself. But in the end, as Rich said on that episode, you're not just endangering yourself, you're endangering other people as well. It's very difficult. I totally understand it.
Every time I think about this, I try and, it's easier for me. I'm older. My circumstances are different. But this is not going to last forever, okay? I basically encourage patience, okay? I know it's hard. But, a year for me isn't as much as a year for, a 17-year-old. I understand. But it's not going to last forever.
I just read an essay by somebody in their 80s, who said, this is taking a year from my life, and I've heard other people say, I'm going to have to shorten my bucket list. So you may be better at being patient than some others, so.
I don't know. I don't feel like I've been robbed of a year. I've experienced a pandemic. This is amazing.
It's a good way to look at
it. I agree,
yeah.
But everyone's different. And our son asked me, how long till I get vaccinated and I can go back? And I said, late spring, early summer. He was devastated, right? So people just react differently. Mm-hmm. And you and I, we can just do this, but we have we have a network that we are depending on. Yeah. I realize I have this podcast network that is probably sustaining me more than I realize, right? And so I can't really shrug it off and say, I'm okay in my basement. That's not right. But
go ahead.
Go ahead. No, I'm done.
I was going to say, to address the messaging question, it might not be in the bailiwick of epidemiologists per se, but public health people could potentially do experiments to figure out what are some of the best ways to do messaging. I think there's a long history of that with respect to, cigarette smoking, other, risky behaviors and things. So, maybe there'll be more research along those lines in the future as well.
I'm sure. All right, one more. Rich, can you take that last one?
Keith and Carol write, howdy and happy holiday. Howdy all and happy holidays. Thanks for all you do. It means a lot. I started viewing Vincent's virology course on January 2nd, 2020. How prescient. And highly recommend it to all TWiV listeners. With all the changes this holiday season, our family decided instead of being gloomy about everything that is happening, we'd instead include this tiny non-living particle that has had such an impact on our lives and, in a tiny sense, own it. This is Coronavirus Christmas. So she's singing my song. Included are a couple of pics of our SARS-CoV-2 tree, which we named Vinny the Virus. In the spirit of itself, while making it, we were not allowed to go outside to get any supplies and had to make do with what was lying around the house. Everyone had to wear sweatpants and no one was allowed to shower. I think my beard grew 3 sizes that day. We are particularly proud of both the S and M proteins and the genetic variants we gave to them. After finishing, we stood there admiring our work, and my kid said, "Too bad the virus wasn't this big in real life. At least we'd seen it coming." Truer words could never have been spoken, and that is why it behooves all of us to physically distance, mask up, wash our hands, and simply assume that everyone, including ourselves, are carrying these terrible and wondrous buggers inside us so that we can all be around next Christmas to celebrate with our loved ones that we did the right things and kept each other alive and well. All the best to everyone. Let's all be safe. Keith and Carol and family. By the way, it's 1C and looking like snow. And sends a picture of their COVID Christmas tree. My goodness. Look at this. It's amazing.
Very nice.
They used all stuff around the house. I'd have to blow this up and see what's there. But there's this great giant SARS-CoV-2 with strings of, as if it were a tree topper.
Yeah.
But it's
Very cool.
most of the deal. Hanging
Nice.
from the ceiling with strings of lights coming down from that to make it look like a tree. This is phenomenal. Thank you.
Yeah.
You know, there's another group of people who react differently, right? We're going to make
Yeah.
the best of it.
Right.
Yeah.
Everybody's different. I think it's pretty cool you decided on January 2nd to start taking my course. That's a pretty good time to start.
Yeah.
All right. Let's do some picks. Brienne, what have you
I
today?
have an article that I have been sharing with quite a few people of late. This has been one that a lot of my colleagues have enjoyed when I've shared it with them as we're thinking about going back to teaching. So this is, I found it from Lindsay Marr, who is a engineer at Virginia Tech who works on aspects of aerosol viral transmission. And she is the co-author, along with Monica Gandhi, of an article in Cell that was published in mid-December that is titled Uniting Infectious Disease and Physical Science Principles on the Importance of Face Masks for COVID-19. And this article talks quite a bit about some of the studies that have shown positive effects for masks, but also talks about recommendations for masks for members of the public and which types of masks are particularly effective for the public. And there are all sorts of citations, so you can look at the data about all of this, both in terms of thinking about physical science and in terms of thinking about infectious disease. And I was sort of excited to see some of the figure here where they show types of masks that lead to maximal protection that can be used sort of by an everyday person. I was excited to see that the choice that I have been going with throughout this pandemic was one that was listed for maximal protection. But it did also really helped me to answer questions from people who have been wondering about what might be good to do for masks. And so I found this paper and all of its references, being there nicely in one place to be incredibly helpful in answering questions.
Yeah, it's good.
Cool.
Good to send to people. It seems to be clearly written, so most people can get it. Yeah.
It really is, and everyone has seemed to appreciate it when I've sent it to them.
Good. Very good.
She's quoted in the recent Ed Young paper as saying, I've seen more acceptance in the last 6 months than over the 12 years I've been working on this about viruses being transmitted by the air. And I think it was in here. No, that's it. Yeah. Okay.
'Twas COVID in the aerosols.
Yes.
Rich, what do you have for us?
This comes from my grandson, Porter, okay, who was all excited about this and wanted to show me this video about how if you cut a grape almost in half, but not pull it apart completely, and stick it in the microwave and turn it on, you can get plasma, okay? So it gets so hot that it ionizes to form plasma, and you get these big flare-ups, okay? And what he showed me was a video from a YouTube channel called Veritasium that I'll get to in a minute. And the video is like supremely nerdy. It's wonderful because it starts off with this thing about making plasma in the microwave from grapes. And apparently this guy who runs this YouTube channel published this some time ago, and a couple of physicists saw it and got on top of it and elaborated on it and published a PNAS paper about how this happened. And this is a follow-up YouTube video with an interview with these two physicists who published the PNAS paper, elaborating on why it is that when you microwave a bisected grape, you get plasma. Okay, so I'll leave that to you. Not only is it cool, but it's nerdy in the extreme. But as I dug into this YouTube channel, Veritasium, And if you look at the front page of this, it's the author, and it's Derek Muller, or Muller, M-U-L-L-E-R. Has a little video where he talks about how the YouTube channel evolved. And he's a person with engineering training, but his real passion is filmmaking, and how he turned that into basically his own career as a YouTube guy. He has 8 million subscribers, okay? And a bunch of really cool, nerdy science posts. So there you go.
That sounds really cool. But the question that I have for you, Rich, is have you tried this with your grandson?
Ooh, no, I have not. And I don't even have any grapes lying around. But yeah, I think we'll have to do it when nobody else is home. I might get a little pushback from others about this, but yeah, that's a great idea.
Okay, I won't tell them.
You can get some grapes, yeah. Veritasium. Very good. Yeah, that's how you get 8 million subs. You do wacky things like this.
Well, I mean, he obviously
and with the background.
Yeah.
Kathy, what do you have for us?
So, as is often the case, I picked an astronomy picture of the day, and it happens to be today's, and it's striped sand dunes on Mars. And they talk about dry ice sublimating, and when it usually happens. But this picture was taken in the autumn on Mars, and so they don't really have a good explanation of it. And it's just a cool picture. The thing that it's a little frustrating for me is that there's no scale on it. So I have no idea how big these things are. But maybe with a little more research, I could figure that out. But anyway, it's just a very cool picture. So check that out.
Weird.
The caption below has lots of links, and you could potentially maybe figure out more about these than I had time to do. And the second thing is that follow-up to last week's picks about Auld Lang Syne. First of all, I was a little bit hesitant when I announced where the choral group was from, and it was indeed from University College Dublin. And Vincent fixed that in the links on the webpage, but I wanted to also say that here. That group has an interesting way that they did the filming of that video, and it's quite spooky at the very end, I think. But then I had, and I think Vincent got this too, email from Laura from Texas, another Auld Lang Syne that is very powerful as well for 2020. And so I encourage you to take a look at that one. That's another 3 or so minute one, in addition to that University College Dublin one, in addition to the longer one by John Green that I picked last week. And then those of you that heard Daniel's clinical update, he started out with his quote of Auld Lang Syne, which I think he may or may not have done before he heard my picks. So, I think that one way or the other, we didn't know about each other's picks. So it's just coincidence.
Very cool. My pick is a little essay by Paul B. Nash, a virologist who has been on TWiV at the Rockefeller University. He had put this on Twitter earlier this week, and I thought, you should have this in a proper forum, and so I posted it at Virology blog. And it's basically, he says, we can make vaccines that work by by making antibodies against spike. That's all cool. But, if I were nefarious, here are some things I would do to make the vaccines impotent. And he lists a bunch of things that we're doing, like delaying rollout of testing, keeping schools open, starting a rumor mill, treat people with untested convalescent plasma of unknown potency. And then he goes, here's the kicker. Administer vaccines to millions of people, but delay the second dose, which is something we talked about today. Of course, he ends, I don't know if these would be successful, but that's what I'd try if I wanted to generate vaccine-resistant SARS-CoV-2 variants. And it's really well done. So check it out. It's relatively short. All right, that'll do it for TWiV 703. You can find the show notes at microbe.tv/TWiV. Questions and comments, go to TWiV at microbe.tv. And remember, if you want Daniel to answer your question, it's Daniel at microbe.tv. Because if you don't do that, I will never see the Daniel email. So Daniel at microbe.tv for clinically related stuff. Everything else, TWiV at microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Kathy Spindler's at the University of Michigan, Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thank you, Brianne.
Thanks. It was great to be here.
I am Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back in a couple of days. Another TWiV is viral.